Okay, good afternoon, class. This is the video discussion for the second part of your chapter 5, proteins. So this is quite a long topic and I will divide the discussion for this into two parts. So this is the first part. So let's start with the formation of polypeptides. Okay, so how are your polypeptides made? So proteins are made up of amino acids connected through amide structures. Again, your amide structures are what? These are amino or these are structures containing a group of nitrogen. Okay? So, ang structure niya is, tingnan niyo yung structure ng inyong amino acids and it contains the nitrogen or amino group. Okay? So, all in all, that is the amide structure. So every time an amino acid is joined to another amino acid, water molecule is eliminated. So tingnan nyo dito sa example. In order to connect or to bond two units of amino acid, then you will remove a molecule of water. Okay? Kaya dito sa product niya, you will see one molecule of water or H2O. So, the bond that connects one amino acid to another is called your peptide linkage. Okay? So, your peptide linkage, this is the linkage happening between the carboxyl group of your first amino acid and the amino group of your second amino acid. So, ito siya. You will see here, this one. This is the carboxyl group and it will react with the amino group of the second amino acid. So that is what you call the connection between these two, or the connection that will form between these two is what you call the peptide linkage or the peptide bond. Okay, so the hydroxyl or the OH coming from the carboxyl group of one amino acid reacts with the H or the hydrogen in the alpha amino group of another amino acid. So, bakit alpha amino group? Because the amino group is bonded to the alpha carbon atom in your amino acid. Bakit siya alpha? Because it is the first carbon atom na nakadikit sa iyong carboxyl group. Okay? So, yung OH group ng first amino acid will react with the hydrogen of your NH2 sa second amino acid. Okay? And by this process, you will eliminate a molecule of water. For example, we react two molecules of valine. The product formed is called valine. Okay? Or in short, yung kanilang shorter term, you have val. Okay? So don't forget when you write class, the shorter name or the shortened name of your amino acids. Ito kasi yung longer name niya. Ito yung buong name. And then kapag pinasulat sa inyo yung abbreviation na name, don't forget to place eto. Yan. Baka makalimutan nyo na naman, ha? So, always yan. So, because this symbolizes the peptide linkage. Okay, so highlight ko na. Okay, so valine is called a dipeptide because it consisted of what? Or it consists of two amino acid units. So dipeptide. Okay, so ito yung example niya. You have valine and you have valine. Okay, so yung carboxyl group niya, ang OH, will be reacting with the hydrogen of your amino group, forming a molecule of water. Now, when you connect them, okay, so sa linkage nila, eto na siya. So, nagiging NH na lang, okay? NH na lang yung natira sa NH2 kasi nga, kinuha na yung hydrogen and sa carboxyl group CO na lang kasi kinuha na yung OH. So, idugtong nyo lang kasi madali lang naman siya. And then, nakita nyo kasi dito sa side na to, nasa left pa yung R group niya or alkyl group. Okay? So, para hindi kayo mahirapan, you can place this group, ito, on top of your alpha carbon atom. So, pwede nyo siyang ilagay sa taas so that madali nyo lang makakonect yung inyong N with your carbon sa carboxyl group. Okay, so you can place that on top and you can actually make your double bond in a slanting position. Okay, so yan siya. Example nito. So if there are 3 amino acid units, you call that as tripeptide. Okay, kapag 4, you call that as tetrapeptide. If 5, you will call it as pentapeptide, and so on. If there are 40 to 50 amino acids in the peptide chains, then they are called polypeptides. Okay? So, less than 10, pwede pa siya, you can place the name. Pero kung more than, or let's say 40 to 50 amino acids na, yung nandoon sa inyong peptide chain, then you call that as polypeptides. Okay? So, amino acids, and then you have polypeptides, and then you have your proteins, okay? So, ang proteins, yun na siya yung combination ng iba't ibang units ng polypeptide, okay? Yung pinagkaiba niya. Your polypeptides are composed of amino acids, while your proteins are composed of units of polypeptides. Okay, so longer chains of amino acids are called proteins. The amino acids in peptides without water are called amino acid residue. So, when you combine two or more amino acids, diba, you will a molecule of water will be eliminated. So, yung mga those amino acids na na-remove na ang kanilang water, yung OH and H, you call them as amino acid residues. Okay? So, ito na. Kita nyo ito. This one and this one, these are amino acid residues. Meaning, these are amino acids na wala ng water molecule. Okay? So, when a polypeptide chain is made, a carboxyl group and an amino group are involved in each peptide. So, adjust ko lang kaunti. So, masakit ng likod ko. Okay. So, again, when a polypeptide chain is made, a carboxyl group and an amino or an alpha amino group are involved in each peptide bond. So, actually, class, you can also call the peptide bond as amide bond. Bakit? Kasi yung amino group is connected to a carboxyl group. Okay? So, your amino group is involved in the linkage. Kaya pwede mo ring tawagin na amide group or amide bond yung inyong peptide bond. Okay, so the groups linked by peptide bonds cannot ionize as acids or bases. Bakit ano ba yung pwedeng mag-ionize into acids and bases? Yung inyong amino acids that are free, okay? Katulad ng zwitterion. So, di ba, yung zwitterion ninyo is very reactive because it contains the positive and negative charges. Kaya, kapag it can act as an acid or base depending on what solution is present, either an acidic solution or a basic solution. Pero kapag yung amino acids ninyo are already connected by peptide linkages, di ba? So, wala na siyang charges and therefore, it cannot ionize as an acid or base. So, the properties of polypeptide chain or protein are determined by the side chains of the amino acid residue. So, again, yung amino acid residue, diba? Siya na yung natirang amino acid or the amino acids that does not contain a water molecule. So, for example, itong valine natin. So, saan ang side chains nila? Remember, the side chains, ito yung mga varying alkyl groups na nakakonect. Okay? So, dito, CH2CH3CH. Yan ang inyong side chain. On the other hand, dito sa kabila, ang side chain mo is CH, CH3, CH2. So, Actually, same lang sila kasi valine naman silang pareho, okay? So, remember, ang side chain natin, yan yung alkyl groups or yung straight chain or straight carbon chain. Okay? So, varying yan. Pwedeng magkaiba. So, tingnan nyo yung table number 5 doon sa first part. Diba? So, makita mo doon yung iba-ibang types ng side chain. Okay? Yung R group doon. So, yun ang side chains natin. So, the side chains that are present in your amino acid residues will determine the properties of your proteins or polypeptide chain. Okay, so one end of the polypeptide chain, okay, has a free amino group and the other end has a carboxyl group. So remember, in a polypeptide chain, always kang merong dalawang ends. Okay? Yung isa is a free amino group, meaning ang end na yun is containing amino group or meron siyang nitrogen. On the other hand, yung isang end, meron siyang carboxyl group or C double bond oxygen, okay, or carbon double bond oxygen. So the amino group end is at the left and called N-terminal residue. Okay, so itong nasa left side, this is what you call, for example, itong met, okay, or methionine. So ito siya ang N-terminal residue because ang present sa kanya is, or ang group na present sa kanya is a free amino group. On the other hand, yung nasa right side na end, you have your leucine here. So it is actually called as the C-terminal residue. So in short, carboxyl group N. Okay, so tandaan niyo yan siya on the left side always N-terminal residue and on the right side always C-terminal residue. Okay, so the numbering starts at the N-terminal residue and ends in the C-terminal residue. So in naming, all the amino acids from N-terminal residue ends are named by changing your INE. Okay, so if napansin nyo, lahat ng amino acids natin, they end in INE. Okay, except your glutamic acid. Okay, yung may dalawang acid natin. Okay, you have two amino acids that ends in acid. Okay, so you will change their names into from INE you will change it to or to YL, ending and only the last amino acid in the C-terminal residue will not change its name. So lahat ng amino acids starting from the left side hanggang sa pinakahuli, okay, or hanggang sa amino acid na katabi ng pinakahuling amino acid, then you will change the end of its name from INE to YL. That's why kanina dito sa example, you have valine. Okay? So, two valine amino acids. So, yung nauna, the one on the left side will be changed to valine and yung ending or your C-terminal residue will stay the same. Okay? The name stays the same. So, in short, if you have more than two chains of amino acid, for example, methionine, prolyl, glycyl, tyrosyl, lalanyl, and lysyl and leucine. Okay, so yung pinakahuli, the one on the very end will remain the same. Okay, so as is yung kanyang name, and then lahat ng nasa left side niya will be ending in YL. Okay, so with two amino acids, they can form two different dipeptides. So, iba-iba, we have actually different sequences, okay, for different numbers of amino acid units. So, for example, you are given with two different types of amino acid. So, dipeptide siya. You have the valine and the serine. So, pwede kang may dalawang two sequence. Pwedeng first ang valine ang mauna, followed by your serine. Or pwede ring serine ang mauna, followed by your valine. Okay? So, pwedeng valylserine or valser, or pwede ring serine, valine, or serval. Okay? So, you will use probability. Okay? So, if I think you've encountered already sa mathematics yung probability, how to solve the probability. This is easy class. You just use your calculator, okay, scientific calculator. So how many combinations are possible when three different amino acids react? Like for example, methionine, alanine, and tyrosine. So using your probability, so 3, using an exclamation mark, is equals to 3 times 2 times 1. Okay, so equals to 6. So meaning, you will have 6 different sequences for the combination of 3 amino acids that were given. Okay, so meron kang 6 na tripeptides, na possible tripeptides that will be formed from the combination of your methionine, alanine, and tyrosine. So each amino acid appears only once. These tripeptides are, so ito siya, met, ala, and tyrosine. Okay, so methionine, alanine, and tyrosine. Methionine, tyrosine, alanine. Alanine, tyrosine, methionine. Alanine, methionine, tyrosine, and so on. Okay? So, if tatlo lang, if smaller units yung ginamit, siguro pwede mo pa siyang i-mano-mano ng solve. Okay? Pero kapag sobrang dami na, mas malaki na yung magpo-produce mo na probability. So, it's better to use calculator in this. So, paano siya? You press the shift on your calculator. I-press niyo yung shift and then press the x to the power of negative 1, I think. Wait, I will get my calculator. Okay, so ito class, you will see here, eto siya. Yung shift, ayan, and nasa baba niya lang yung other button that you will press in order to get the exclamation point. Okay? So, nakita niyo ba? Ayan. This one, shift, and this one. Okay? So, for example, 3, okay, may symbol na rin naman pala dito, class. X, exclamation point. So, makikita niyo rin. So, 3, probability. Ayan. So, I don't know if nakikita nyo. I think hindi. Pero yun, effective siya. So, you just use that in calculating for the probability of the possible sequences. Okay? Na pwede sa or the possible sequences for the combination of certain numbers or units of amino acids. Okay, so the number possible, let's go back. The number possible peptides increases rapidly as the number increases. So, mas tumataas yung number ng units ng amino acids in a polypeptide chain, mas tumataas din yung possible or probable sequence na pwedeng mangyari doon. Okay? So, for example, if there are 5 different amino acids that will react with each other, so 5, okay, exclamation point, then you will have 5 times 4 times 3 times 2 times 1 is equals to 120. Okay, or probability of 5 units of amino acids will result to 120 possible sequences. Okay, so different combinations of pentapeptides using each amino acid only once. So if there are 15 different amino acids reacting, it produces a combination of more than 1 trillion, okay, possible sequences. So you can imagine the number of different combinations for 20 different amino acids which are produced. Paano na lang kaya if 40 to 50 and more than 50 na yung amino acid linkages, di ba? So, ganon ka-broad yung range. Okay? ng sequence na pwede mong makuha from your proteins. And that is one of the greatest and most unique characteristic of proteins. Okay? Kaya yung functions ng proteins niyo napakalawak din. Kasi 'di ba, ang dami-daming trillions and maybe billions pa nga ang pwedeng maging sequence, and each sequence will produce a certain type of protein. Okay? And may iba-iba rin silang function. So, that shows how big your proteins can become. Okay? Depending on the number of units of amino acids present on its structure. Okay? So, there are naturally occurring polypeptides with significant biochemical functions. Okay? Naturally occurring meaning, ano na siya? You can find these polypeptides sa environment, whether rin sa organisms, okay? So, produced by certain types of organisms. So, at present, over 30 different polypeptides are known. In general, they serve as hormones or neurotransmitters. Yun yung main function nila. Hormones for producing different processes or reactions in the body, and neurotransmitters, which function as controlling pain and pleasure response in the brain, is to controlling smooth muscle tissue contraction and or rate of kidney fluid excretion. Okay, so yung natural polypeptides na yun na nakikita in the environment and in other produced by other organisms, they usually work as what? As pain or controlling pain. So, usually sa medicine ito sila, medical uses for opioid effects. Okay? So, analgesics, mga pang-control ng pain. So, the polypeptide's biological effectiveness and specificity depends on the amino acid sequence and chain length, yung haba ng amino acids. Okay, so for example, there are two naturally occurring polypeptides which affect the brain as pain relievers. Okay, so itong dalawang naturally occurring polypeptides na ito, for pain reliever ang function nila. You call them as, okay, ito, leuenkephalin ito. Okay, palitan nyo ng H sa copy ninyo. Leuenkephalin, enkephalin, and metenkephalin. Okay, so if the amino acid sequence is changed, it results to a different side chain characteristic, causing drastic changes in their analgesic effects. So, pwedeng nawawala yung function nitong dalawang naturally occurring polypeptide, leuenkephalin and metenkephalin, kapag may nagbago sa amino acid sequence nila. Okay? So, the substitution of L-alanine for either glycine residues reduces 1,000-folds in their effectiveness as painkiller. So, kahit isang L-alanine lang yung mapapalitan ng glycine doon sa sequence ng inyong leuenkephalin and metenkephalin, it can affect thousands of the effectiveness of these painkillers. So, substitution of L-tyrosine. Okay, kung papalitan mo naman yung L-tyrosine sa sequence niya for L-phenylalanine, this will cause a comparable loss of activity. Okay, so, nawawala yung activeness ng dalawa. And also, changing L-tyrosine to D-tyrosine causes a considerable loss in analgesic effectiveness. So, isa lang yung point doon, class. Kahit isang unit lang ng amino acid ang pinalitan mo doon sa sequence nitong leuenkephalin and metenkephalin, nawawala yung kanilang effect as painkillers. So, it is clear that a particular amino acid sequence is essential for proper polypeptide function, whether the side chain is large or small. So, hindi mag-matter if yung side chain mo is very complex or very simple. Okay? Again, ano yung side chain? The alkyl side chain of your amino acids. Okay? So, polar or non-polar man sila. Okay, when you say polar, very reactive in water, lalong-lalo na sa water. Or non-polar, meaning wala siyang positive and negative charges and therefore it is not reactive to other substances. Acidic man siya, basic or neutral, so they should be in their proper position. Okay? Kasi kapag nagkaroon ng interchange sa position ng sequence ng amino acids in a polypeptide, then it will result to losing their specific function or effect. So take a look at oxytocin and vasopressin, which differ at 2 amino acids. Okay, so their biological functions differ dramatically. For example, ang vasopressin natin, this raises the blood pressure, used to treat surgical shock, antidiuretic din siya, and its absence leads to diabetes insipidus, a condition excreting up to 30 liters of urine per day. So, ano kasi class, when you say antidiuretic, diba, ito yung it maintains the level or the volume of body fluids, usually by using urination. Okay? So, ayan. Kapag walang vasopressin, you will not be able to excrete your urine. Hindi siya malalabas. So, may ipon lang siya sa loob ng katawan mo. Okay? So, ang nangyayari dyan, mas dumadami yung urine mo per day. Okay? So, 30 liters of urine per day is not normal, ha? It is abnormal. So, yan yung cause ng diabetes insipidus. So, dapat merong reabsorption, okay, ng fluids in the body para makontrol mo yung body fluids. And yun yung ginagawa ng antidiuretic hormone. Okay? So, vasopressin. So, vasopressin can control this condition. Oxytocin controls contraction of uterine during labor. Okay, so kapag walang oxytocin or kulang yung oxytocin during labor, nahihirapan ilabas ang baby. Okay? Kasi hindi magko-contract yung walls ng uterine mo to push out the child. So, it causes contraction of the smooth muscles also in the mammary glands for milk excretion. Usually, yung function ng oxytocin, may pinapalabas siya. Okay? It causes contraction in certain muscles, either in linings of the wall or in muscles or smooth muscles, to produce or to secrete something or may ilalabas ka. So, oxytocin and vasopressin were isolated and synthesized by Vincent Duveg. Kalimutan ko yung pronunciation niya. Basta yan na siya, si Vincent, and co-workers at Cornell University. He was awarded the Nobel Prize in Chemistry in 1955 for this work. Okay, so ang ginawa niya, in-isolate niya sa certain or coming from an organism, okay, or synthesize ito class. So, he produced in the laboratory yung oxytocin and vasopressin hormone. So, there is no difference between the synthetic and natural material. Okay? So, synthetic man or nanggaling talaga sa organism or in the natural environment, yung oxytocin and vasopressin is the same lang ang effect. Okay? So, it is commercial use to induce labor in the late stages of pregnancy. So, you have here in table 7, these are the biological polypeptides, primary structure and function. So, class, ito yung shortened name niya. Okay? This is the name of the polypeptides that are abbreviation. Okay? Abbreviation yung gamit. So, again, remember, don't forget to write yung kanilang dash bang tawag dyan. Okay? Para to represent or symbolize the peptide linkage. So, kapag may nakita ka class, when you see this, yan, this means cyclic ha, meaning your cis or your cysteine and another cysteine is bonded to 2 sulfate, okay? So, ang tawag mo dito is disulfide linkage between 2 cysteine amino acid units. So, kapag sinulat mo yan yung structure niya ng buo talaga, mag-form siya ng parang cyclic, okay? Kasi kinonect mo yung dalawang cysteine na amino acids on the left and the right side by a disulfide bonding. So, by the way, your disulfide bonding is an example of a covalent bonding. So, explain ko yan mamaya. What is covalent bonding? Okay, so, you have here, biological polypeptides, ito sila lahat, class. Okay, so, napansin nyo, they are composed of less than 40 to 50. Okay, less than. Polypeptide siya kapag less than 40 to 50 pa. Okay, kapag more than dyan, then proteins ang tawag doon. So, these are examples of polypeptides with their primary structure and their general biological function. So, you have substance P. Ang function niya for pain-producing agent. Okay? Bradykinin. Okay? So, or bradykinin, you have its function is it affects tissue inflammation and blood pressure. Your angiotensin number 2 or angiotensin 2 maintains water balance and blood pressure. Okay, ito ng dalawa. Leu and methamphetamine relieves pain and produces sense of well-being. Vasopressin, ayan na, increases blood pressure, decreases kidney water through excretion. Oxytocin initiates childbirth, causes mammary gland milk release, affects kidney, ganun din. Thyrotroponin, releasing hormone. So, ito siya, tatlo lang, maliit lang. Tatlong units lang ng amino acids. So, you have, it stimulates release of hormones from the pituitary gland. Orexin, so, hindi na sinulat kasi ang haba-haba, 33 amino acids long. It causes wakefulness. So, ito dapat yung meron tayo kapag when we are studying, okay, orexin. I don't know where you can find that. May mabibili bang medicine that contains orexin, but don't take that class drug na ang tawag dyan. Okay, so, orexin for wakefulness. Neuropeptide FF modulates pain sensation. Neurotensin involved in brain memory function. Okay, neurotensin, ito yung usually nakikita mo sa mga brain supplements, yung mga supplements for memory. So, yan siya. Neuropeptide Y stimulates eating. Okay? And then endomorphin acts as morphine-like analgesic. When you say analgesic class, this for pain reliever siya. Okay? Okay, write the structure of a tripeptide, valine, serine, and thionine, and give its name. Okay, so tatlo. So kapag ganito pa, sundin nyo lang yung, kapag hindi niya naman hiningi yung iba't ibang klase ng possible na sequence, so sundin nyo na lang yung binigay na pagkakasunod-sunod. So mauna ang valine, sunod ang serine, and then last ang methionine. Okay, so first, isulat mo muna. Always remember ha, para meron kayong points sa activities. Kapag ganito, you write first, okay, the structures of the given amino acids. Isulat mo muna on top, then after that, show the linkages that are formed. Okay, so free amino acid group is at the left, and the free carboxyl group is at the right. So remember, dito sa sample natin, meaning, dapat meron kang free amino group or NH2 sa left side, sa valine, and meron kang free carboxyl group, COOH, sa right side, sa methionine. Okay? So, remove the water molecules between carboxyl group of valine, dito, and amino group of serine, eto. And between carboxyl groups of serine, this one, and amino group of methionine, this one. Then connect the carbon to nitrogen to form the peptide bonds. So, ayan na. You remove the water molecules in order to form the peptide bonds. So, ang natira na lang is carbon, double bond oxygen, and dito sa side, sa serine, and H. And then, dito naman sa right side ng serine, so wala nang natirang naalis yung OH, you will be left with carbon, double bond oxygen, and the NH group of your methionine. So when you write the name, don't forget to write the name in your activity or quiz. Valyl serine methionine. So only the methionine remains with its original name. Okay? Kasi nasa C-terminal residue siya. Okay. Let's go to protein structure and function. Okay, so this class on, the protein topic is more on memorization. Okay, so wala siya masyadong writing of structures. Unlike yung una, this is more on memorization and familiarization. Okay, so try to memorize kung ano yung mga functions ng polypeptides and yung mga important details lang naman na nakasulat or included dito sa materials inyo. Okay, so let's start with protein structure and function. What are the different structures? And ano yung function ng specific structures na yun? Tinanong ko ito sa question and answer ninyo. Okay, for a protein molecule to serve a specific biological function, it must have a defined overall shape. Okay? So, chemists describe large shape of proteins based on the following structural level. So, may iba-iba kang, you have 4 different types of protein structure depending on their complexity. Okay? Kung gaano sila ka-complex, kung gaano sila kalaki, and kung gaano sila ka-simple. Okay? So, yung pinaka-simple natin is your primary structure, followed by your secondary structure. Next is your tertiary structure, and ang pinaka-complex sa lahat and ang pinaka-large din, you have your quaternary structure. Okay, so primary structure, coming from its name, this is the starting point of the formation of a protein molecule. Okay, so your primary structure is actually the most important structure out of the 4. Why? Because if there's something wrong with your primary structure, kapag sa primary structure pa lang, meron ng mali sa sequence, then the whole protein, yung buong structure ng protein will be, ang function niya will be different, okay, from the original dapat na function. Okay, so masisira yung buong protein mo kapag sa primary structure pa lang, meron ng mali sa sequence or may kulang na amino acid unit sa sequence. So remember, even though your primary structure is the simplest out of the 4, it is actually the most important. Okay, so your primary structure is established by the number, kind, and sequence of amino acid residues, which compose the polypeptide chain or chains making up the molecule. So it determines the alignment of side chain characteristics. Kasi sabi nga natin, your amino acids contain side chain. Ano yung side chain? Yung nagbabari na R group. Okay? So, pwedeng ang R group mo is alkyl group. Ano pa yung ibang types niya? Nabanggit doon sa unang part ng protein chapter. Okay? So, yung side chains na yan, ang nangyayari kasi dyan, class, yung side chain is very important because nagkakaroon ng bonding, okay, sa different side chains of the amino acid units inside a protein or a polypeptide, okay? So, if meron kang mga, sample natin dito, if meron kang mga amino acid units, for example, ito, amino acid units, okay, nakalink sila. Yung mga side chain nila sa gilid, may mga connection din with other side chains of other amino acids. Okay? So, yung formation ng side chains or the bonding of the side chains of different amino acids actually affects, no, the structure of the resulting protein. So, itong side chains, yan ang nagpapalakas. Okay? It strengthens the structure of a protein. Because the more bonds that are present in the structure of a protein, mas, mas durable, okay? Hindi madaling masira. So, okay. which determines your side chain proteins determines the three-dimensional shape. Okay? Yung 3D shape ng isang protein. Okay? Kasi kapag nagkaroon ng side chain bonding, remember, the side chain bonding produces the 3D shape of your protein folds. Okay? Kasi nagkakaroon ng baliko-baliko doon sa structure ng protein mo kasi nga, nagkakaroon ng bonding sa side chains ng different amino acid units. So that is why the amino acid sequence is very important in establishing your shape of protein. So it is very huge task to determine the sequence of the amino acids even in one protein molecule. So imagine mo na lang class, kung isa kang matalinong scientist at gusto mong alamin ang sequence ng amino acids containing 100, diba? So, diba, napakahirap niyan kasi napakalaking molecule niya. And you will have to check one by one the sequence and dapat wala kang mali. Okay? So, in 1955, ginawa yan ni Frederick Sanger. Okay? So, he determined the amino acid sequence of the beef insulin or the insulin found in the meat of your beef. Okay, so he was awarded dahil sa ginawa niya in 1958 pa yan na hindi pa high-tech pero he made it possible to know the sequence of the beef insulin. So he was awarded with the Nobel Prize for this work. So insulin is a hormone that regulates blood sugar level. Insulin deficiency leads to condition known as diabetes. So paulit-ulit na ito class, hindi ko na yan explain. So, human insulin is made up of, ilan ang amino acid units niya? 51. Okay? In 2 polypeptide chains. So, meaning, yung inyong insulin, dalawa siyang polypeptide chains. Okay? And yung dalawang polypeptide chains na yun, nag-connect, and it contains a total of 51 amino acid units. So, the two chains are joined by disulfide linkages. So, yung dalawang sulfur, okay? So, nakita yung kanina yung binigay ko na sample. Ayan, ito ang isang disulfide linkage. Okay, so, the two chains, okay, yung inyong polypeptide chains, nag-join sa kanila is disulfide linkage or linkage happening between two sulfur ions. Okay, so yung tandaan, ang disulfide linkages will only appear in the cysteine amino acid. Okay? Nakikita lang siya sa cysteine or its linkage or connection only is available to cysteine amino acid or cysteine residues at two different sites. So the primary structure of human insulin is shown in figure 11. Other animals' insulin differs by 1, 2, or 3 amino acid residues in chain A. Okay, so nakita nyo, this is the first chain, chain A. Yan yung unang chain ng polypeptide. Okay, this is the second chain of polypeptide because remember, 51 daw. 51 amino acids ang nagko-compose sa inyong insulin. So, kung bilangin mo, the first chain or chain A is composed of 21 amino acids, diba? So, yan. 1 hanggang 5. Ang dulo niya is 21. Yung chain B mo, composed of 30. So, add mo 30 plus 21, 51. Okay? So, dalawa siyang polypeptide. Kasi magkaiba man itong dalawa na the two are different polypeptide chains. Pero, connected sila. Why? Because of this disulfide linkages, ayan, found in two cysteine amino acids from two polypeptide chains, okay? So, yung cysteine dito sa chain A connects with the cysteine found in chain B through your disulfide linkages. So, dalawang disulfide linkages. Okay? So, these two disulfide linkages forms the structure of your insulin. So, actually, dito sa chain A, meron na rin siyang disulfide linkage, diba? dalawang cysteine. Okay, so that is what you call the human insulin amino acid sequence. So ito yung na-discover or sa human kasi ito. So parang ganito yung na-discover ni Sanger or Frederick Sanger. Okay, so the secondary structure, so primary structure yan siya klasa. Punta tayo sa secondary structure. Okay, secondary structure of protein is the three-dimensional structure held together by hydrogen bonding between oxygen and carbonyl group and the hydrogen of the amino group in the polypeptide chain. So kung kanina sa primary chain, ang included, the linkage that is included is disulfide linkage. Pagdating natin sa secondary structure, nagbago na, no? May nadagdag doon sa linkages. Ano yun? This one, the hydrogen bonding that is happening between your carbonyl group or C double bond O and your amino group, your NH. Okay? So, ayan. Since negative ang O and positive ang hydrogen, so they will connect or they will form a bond. So, you call that as hydrogen bond. Remember, the characterization of your secondary structure protein, it contains the hydrogen bonding between your carbonyl group and amino group. Okay, so there are two types of secondary structures. May two types ka ng secondary structure according to Pauling and Robert Corey in 1897 and 1971. So they received the 1954 Nobel Prize in Chemistry for this work. So ayan. Yung dalawang types ng structure ninyo na secondary structure, you have the alpha helix and the beta pleated sheet. Okay? So, yung alpha helix, this is formed by intramolecular hydrogen bonding. Meaning, within the molecule, yung connection ng hydrogen bonds or yung bonding ng inyong carbonyl group and amino group happens mismo doon mismo sa loob ng molecule na yon. Okay? So, within the molecule, nagkakaroon ng hydrogen bonding. So, ang na-form niya na structure is helix. Okay? Parang yung sa DNA ninyo. On the other hand, kapag ang inyong molecular or the bonding happens in different molecules, so meron kang intermolecular hydrogen bonding between two different molecules and you will form the beta pleated sheet. So these structures are strongly held in particular conformation. When you say conformation, this means shape, okay? Particular shape by the presence of large number of hydrogen. So ito yung dinagdagan ko ng picture class. I will just upload this again. So pwede nyo lang i-download ulit, okay? Yung PowerPoint. Kasi parang doon sa material ninyo or dyan sa course pack, wala siyang picture. So nilagyan ko na lang dito. I inserted some pictures. Okay, so ito yung hydrogen, or this is the hydrogen bonding in the alpha helix structure or secondary structure alpha helix. So, 'di ba? Ito yung continuous chain ng polypeptides. So, ang nangyayari, sila lang din doon mismo sa polypeptide na yun nagkakaroon ng hydrogen bonding. Kaya ang nangyayari, paikot yung kanyang structure. Okay? So, umiikot. Okay? Kasi nga, within the molecule itself, nagkakaroon ng hydrogen bonding. On the other hand, your beta pleated sheet, coming from its name, para siyang sheet na pleated. Okay? Pilo-pilo. May fold siya. So, this is the result kapag yung hydrogen bonding happens in different amino acid molecules. Okay? So, ito yung first chain. Ito naman yung second chain. So, magkaibang polypeptide molecules yan. So, nagkakaroon ng hydrogen bonding within them, causing the structure beta-pleated sheet. Okay, so, let's continue. So, the tertiary structure of a protein is the distinctive the distinctive and characteristic conformation or shape of a protein molecule. So, nandito na tayo. Let's go to tertiary structure. So, if kanina, primary and secondary, ngayon, this is the tertiary structure. Okay, so siya yung nagbibigay ng distinctive shape and characteristic doon sa inyong protein molecule. So this structure is held together by different interactions between amino acid side chains. So kung sa primary, ang bonding is disulfide bonding. Kung sa secondary, the bonding is hydrogen bonding. Dito sa inyong tertiary structure, the bonding is between the side chains, okay, of your amino acid. So, mas complex ito compared doon sa naunang dalawa. So, these interactions are, pwede kang may apat, you have hydrogen bonding. Present sa kanya din ang hydrogen bonding. Number 2, ionic bonding. So, this is the bonding between different charges, positive and negative. And disulfide bonding. Okay, pwede rin sa kanya, may present din sa tertiary structure ang disulfide bonding. And lastly, hydrophobic interactions. Okay, when you say hydrophobic, water feeding. So the following are some examples. Glutamic acid and tyrosine hydrogen bonding. Meaning, nagkakaroon ng hydrogen bonding between your glutamic acid amino acid and tyrosine amino acid. Number 2, glutamic acid lysine ionic bonding. Number 3, cysteine and cysteine disulfide bonding. And phenylalanine leucine hydrophobic interaction. So the number and locations of these interactions which are fixed, strengthen, and the number and locations of these interactions which are fixed strengthen the tertiary structure when the primary structure is synthesized. So, mas maraming present dito sa apat na binigay sa structure, mas malakas or mas durable yung inyong tertiary structure. So, that is why tertiary structure depends on the primary structure. So, sabi ko nga, your primary structure is the most important type of structure of proteins kasi sa kanya nang gagaling lahat. Okay? So, for example, as seen in figure 11, this one, or asan yung figure 11 natin kanina? Eto. So, anong sabi niya? The insulin molecule has 3 locations in the primary structure where disulfide bonding happens. This specificity allows the shape of the insulin. So, tandaan nyo class, yung mga bonding na nangyayari, they are very specific with their location. Okay? So, dapat nasa proper place sila and proper sequence para maproduce mo yung certain type of protein, shape, and structure. So, for example, disulfide bonds are found in the hair. Ang buhok natin, meron siyang disulfide bonds which can be broken by certain reducing agents. Okay? And restored by oxidizing agent. So, this is the key to cold permanent waving of hair. By applying a reducing agent to the hair, some of the disulfide bonds are broken. So, kapag nag-apply ka ng reducing agent sa buhok mo, napuputol yung disulfide bonds. So, the hair is styled with the desired curls or waves. So, for example, straight hair ka, gusto mong magpa-curly hair. So, you will place reducing agent sa buhok mo, and then after that, i-shape mo siya sa kung anong gusto mo na shape ng buhok mo. Either very curly or wavy, ganyan. And after that, you will permanently set this using an oxidizing agent para ma-reestablish yung disulfide bonds. Again, at a different point. Okay? So, ito yung kanyang chart. So, this is the bonds in a normal hair. Okay? May disulfide bond siya. So, you place a reducing agent. Naghiwalay yung dalawang sulfide bonds. And pwede mo na siyang i-reshape sa kung anong shape na gusto mo. And then, you will add, again, an oxidizing agent to reestablish the bonds. Pero, wavy na yung hair mo. Okay? So, pwede ito rin yung ginagamit sa rebound class. Okay? Pareho, the same idea. Okay? Yung sa rebound or rebonding. So, yan yung tinanong ko sa inyo that is the one I asked in your recitation. So, ito yung explanation. By adding, reducing, and oxidizing agent, you will actually rebuild the shape of the proteins found in the hair. So if you look at table 5, for the structures of amino acids, most of them have side chains that form neither hydrogen, ionic, nor disulfide. The uncharged, relatively nonpolar amino acids form the center or the core of most proteins. So remember, kapag uncharged yung protein mo, katulad doon sa sample sa table 5, yung listahan ng amino acids, then these are nonpolar. Hindi sila reactive because they don't have the positive and negative charge. So they usually form yung center or core of proteins. So these amino acids have side chains that do not bond very well with water. Kasi nga, hindi naman wala silang positive and negative charge. And your water will only, non-polar sila, your water will only react with polar substances. So, ang nangyayari, the unsaturated hydrocarbons or lipids form the hydrophobic interactions. So, as a result, yung uncharged non-polar amino acids will turn inward. Okay? Yung mga non-polar amino acid na walang positive and negative charge, pupunta sa loob, okay? Excluding water and forming the core of the protein structure when a protein is synthesized. So the fourth structure of the protein is your quaternary structure. So that is found in complex proteins. So itong quaternary structures, para na ito sa masyadong malalaking proteins. Your complex protein or your quaternary structure is the structure for the whole protein molecule, okay? So these proteins are made up of two or more smaller protein subunits such as polypeptide chains with non-protein components. So, ang inyong quaternary structure, ang components niya ay kumpul-kumpul na polypeptides. So, the quaternary structure is the shape of an entire protein molecule. Okay? Buong protein molecule na siya, determined by how the subunits are held together by non-covalent bonds such as hydrogen bonding, ionic bonding, and other bonds. So, when you say non-covalent bonding class, this is the bonding that does not include the interaction of positive and negative charges, okay? So, hindi siya unlike your ionic bonding na kailangan yung two atoms mo is positive and negative in order for them to interact or react, dito sa non-covalent bonds, hindi na kailangan na ang dalawang atoms mo is may charges. Okay, so ang covalent bonding lang, ginagamit lang siya para i-connect or i-link ang dalawang malalaking macromolecules. Okay, it is used to link macromolecules or two macromolecules that are not containing charges. Okay? So, yan siya. So, example nga natin ng macromolecules is your proteins and what? Polypeptides, proteins, and nucleic acids. So, pa-exercise yan. Okay, so fibrous proteins. They contain highly developed secondary structures. Okay, so ang fibrous proteins mo, ang structure niya is secondary structures. And what characterizes your secondary structures? It is composed of hydrogen bonds. So either alpha helix or beta pleated sheet, yung structure ng fibrous proteins natin. So which means fiber-like sila or elongated shape. So these proteins tend to function in support roles because your secondary structures provide strength. Ang main role kasi ng inyong secondary structure is for support, no? Or kapag sinabing support, mas ano siya? Ano ba yung tamang term? Hindi siya madaling masira, okay? So ginagamit, usually nakikita nyo yung secondary structure ng proteins sa mga bagay na kailangan matigas. Okay? Or sa parts ng body that needs to be hard, okay? For protection. So, one example natin dyan, your example is the alpha keratin found in your hair, the feather, the horns, and the hooves, which depend on the alpha helix for strength. Yung alpha helix kasi natin class, ang mostly, mas compact, okay? So, pansin nyo, the hair, the feather, horns, and hooves, these are all used for the protection of the body. Meron din tayong fibroin or silk protein that is folded into a beta-pleated sheet secondary structure. And lastly, you have the collagen, is the most abundant protein in the animal kingdom. It is a fibrous protein. Okay, so tatlo na yun ha, yung types ng fibrous protein mo. Meron kang fiber-like, meron kang fibroin or silk protein, and you also have your collagen. Okay, so it forms the bone, your collagen forms the bone matrix around in which the calcium phosphate mineral crystallizes. So it composes the ligaments, the tendons, and the skin. Okay, so nakikita sila sa maraming parts ng human body. So this protein structure provides a strong framework for each of these tissues. Collagen is made up of 3 long slender strands, wrapped around another like a rope, which is much stronger than a single strand. So, ang structure ng collagen ninyo is like a rope. So, makikita niyo yung picture mamaya. Okay, so the 3 helical are stacked like cardboard to form collagen fibers, which are aligned to form tendons or deposit as a network to form a bone or skin structure. So, think about the force exerted on your Achilles tendon. Nasaan ang Achilles tendon? Nandito sa likod. Okay, alam niyo naman siguro yan. When you walk and how you are being able to function depends on the strength of collagen. So, another importance of collagen is in artificial biological implants. So, ginagamit ang collagen ninyo as an effective biomaterial. When you say biomaterial, these are materials used in producing artificial prosthetics, pwede siya. So, artificial prosthetics and other artificial bone, skin grafting, and so on. So, nasan ba yun? Okay, so, when you say biomaterials, synthetic or natural materials suitable for use in constructing artificial organs, okay? Artificial organs or prosthetics to replace the bone or tissues. Okay, so ginagamit siya if ever may nag-malfunction sa parts ng body mo, okay, or pwedeng na-break ang bone mo, so you can use collagen in order to replace, okay, bone or body tissues. Okay, so, bakit? Kasi it is suitable or it is stable, it is strong, and have no dangerous allergic reaction. Okay? So, very safe ang collagen ninyo when it is placed or it is subjected to different tissues of the body. Artificial plastics and metals, okay, di ba may ginagamit rin niyang artificial plastics and metals to replace some of the malfunction organs or pwede rin for bone grafting. So, kino-coat sila, no? They are usually coated with collagen before implantation. So, hindi pwede minsan diretso mo na lang ilalagay yung plastic or metal, okay? So, usually, binabalutan muna sila ng collagen, yun nga, para hindi masyado mabigla yung body to foreign materials. Kasi, kilala na nung body yung collagen. So, mas magiging effective yung implants na yun if ever nakabalot siya ng collagen. So, collagen can also be directly used in artificial skin, in bone grafting, and the invascular or tissue sealant. So, pwede siyang kapag may nabutas na or may na-damage na vascular or vessels or tissue, pwede mo siyang ilagay as sealant on top. Okay? Pangtakip. Ayan. So, ito yung structures. Your alpha keratin. This is the structure of your alpha keratin. Okay? So, kapag kinoil mo sa, when you coil alpha keratins, this will be the structure. And kapag you place alpha keratins or single alpha keratin strands in a bundle, this will be, through disulfide bonds, this will be the structure and that will form your hair. Okay? Ito naman yung fibroin. Ang fibroin or fibroin, it is, it forms your silk, okay? Silk protein. So, unlike doon sa inyong alpha keratin, ang structure niya is parang strand, dito is sheet siya kasi composed siya ng beta-twisted sheet, okay? So, silk. And then you have lastly your collagen, which forms your tendons, ligaments, and bone matrix. So, tatlo siyang nag-coil na parang rope. So, yan yung 3 examples of your fibrous proteins. Next, globular proteins. So, your globular proteins have the compact shape due to its own tertiary structure. In the biological world, yan, each unique structure is associated with a particular function. So, globular proteins, okay, it form parang bilog siya, no? Okay? And yung certain structure niya, yung pagiging bilog niya, it serves a particular function in the body. So, some function is to bind specific biomolecules. Pwede mo siyang lagyan sa gitna ng isang biomolecule and it will carry that biomolecule to different parts of the body. So, this is important for biological transport. For example, yung hemoglobin ninyo, it transports oxygen, okay? For processes like immune responses, ang example naman natin dyan is immunoglobulins. Yung immunoglobulins ninyo, it transports certain types of biochemicals for immune function. So, other globular proteins like enzymes, so tatlo na yun ang example niya. Hemoglobin, immunoglobulins, and another one is your globular enzymes, your enzymes. Ang shape ng inyong enzymes is globular protein, bilog siya, that bind biochemical and catalyze the chemical reaction. So, essential siya yan for metabolic reactions. Number 1 is myoglobin. It is one of the simplest of the globular proteins. Ilan lang siya? 153 amino acid residues and 17,000 grams per mole molar mass. So, it is binding protein carrying oxygen for muscle tissue. So, ano yung kinikerry niya? Oxygen molecules. So, a simplified view shows myoglobin to be a globular protein with a folded sausage structure. Okay, unpause. Ayan. So, tandaan nyo, basta sausage structure, folded sausage structure, myoglobin na yan siya. Yan ang keyword ninyo for myoglobin. Sausage, folded sausage structure. Yan. So, in which each section of the sausage represents a segment of an alpha helix. Okay? So, alpha helix din yung bumubuo sa structure ng myoglobin. So, the stability of the myoglobin is due to the alpha helical secondary structure. So, sabi nga natin, ang inyong alpha helix are for strength. Okay? The hydrogen bonds and other interactions between amino acids, side chains, held the folds of myoglobin. So, kapag nagkaroon ng interaction between doon sa amino acids or between sa side chains ng amino acids of your alpha helical secondary structure, you will form the folding of your sausage structure. So, in the center of the structure, there is a rectangle solid wherein iron atom is bound. Okay, so, nakatago doon sa loob ng myoglobin ninyo ang heme group. when you say heme group, this is the portion in which dumidikit yung iron or iron atom, okay, ng inyong hemoglobin. So, heme group coming from hemoglobin. So, may iron part siya sa gitna. So, this part allows myoglobin to store and transport oxygen. Okay? So, saan nagbabind yung oxygen? Doon sa iron part found at the center of your myoglobin. Doon sa heme group. So, myoglobin is conjugated protein because it contains a heme group which is other than amino acid residue. So, dalawa kasi, diba? Simple protein, when you say simple protein, this is a protein that only produces amino acid products when hydrolyzed, meaning ang components niya amino acid lang lahat. Kapag conjugated protein, meaning may iba siyang groups na included sa structure niya. And your myoglobin is considered as a conjugated protein kasi besides amino acids, meron siyang iron group, okay, sa gitna. May iba siyang group na nakadikit sa kanya besides amino acids. So, the improper processing of your heme group will result to disease called jaundice or yung yellowing of the skin. So, bilirubin forms as the heme is degraded. Kasi, tandaan, yung heme group mo, that is the one that causes the redness. Okay? Sa blood mo, that is the one that causes redness. So, kapag nagkaroon ng degradation in the heme group, wala na masyadong oxygen, okay, na makukuha yung inyong myoglobin. Kasi wala na yung center niya na heme, and therefore, more bilirubin will be formed by your liver. Okay, so if the liver does not continuously excrete this yellow pigment, bilirubin builds up in the body tissues, causing yellowing of the skin. Okay? And may indicate liver malfunction. So, ayan siya. This is the sausage, diba? Tama naman siya. Sausage yung kanyang form, and then on the center, you will find the heme ring in which it is the site where your oxygen binds. Okay? So, ito yung skeletal structure niya. Ito naman yung space-filling model na myoglobin. So, marami itong dalawa na to, siguro mas malapit yung totoong itsura niya dito sa space-filling model. Yung skeletal structure niya, ginagamit lang yan to show you, parang bigyan kayo ng idea kung ano yung sausage structure. So, itong skeletal structure, wala pang oxygen na nakabind sa myoglobin, okay? While sa space-filling model, yung maliliit dyan na below, those are oxygen molecules that are binded because of the presence of the heme ring sa center. Okay. So, you also have the carboxypeptidase. A. It is a small enzyme. Ilan siya? Mas malaki siya compared to your myoglobin, composed of 307 amino acid residues and 34,500 grams per mole molar mass. So, it catalyzes protein digestion in small intestine. So, ang main function, kung ang myoglobin ninyo is for transport of oxygen, your carboxypeptidase is for digestion or catalyzes, para siyang enzyme that catalyzes protein digestion in the small intestine. So, it is also a globular protein, bilog din siya, with a ribbon structure. Kung kanina sausage, yung myoglobin, your carboxypeptidase is characterized by a ribbon structure, okay? So, the alpha helix is represented by a curling line. Ito siya, yung curling line dyan, that is your alpha helix. And, ang carboxypeptidase, hindi lang alpha helix ang meron siya. It also contains beta-pleated sheet, which is broader in line, okay, with an arrowhead at the end to show the direction of the protein chain. So, ito yung example. You have the alpha helix, and then a beta-pleated sheet, and then alpha helix na naman, and then beta-pleated sheet. Mas makapal kasi yung beta-pleated sheet mo compared sa alpha. helix. So, parang ganito yung structure niya, ribbon-like. So, alpha-beta pleated sheet or beta-pleated sheet provides a stable core for carboxypeptidase, which is often called a fan structure to describe a twisted beta-pleated sheet. Okay? So, remember, kapag fan structure, it describes the structure of your beta-pleated sheet. So, this is surrounded by a set of alpha helixes to further strengthen the protein structure. So, ginagamit nga yung alpha helix for support. Okay? Your beta-pleated sheet serves as the core, and then your alpha helix structure serves as a support. So, this is surrounded by a set of alpha helixes to further strengthen the protein structure. Proteins share common skeletal structures. Myoglobin and other protein use alpha helix as a spiral tube twisted around to support the bulk of protein framework, while other proteins naman use beta-pleated sheet to provide rigid protein framework like in carboxypeptidase A or the fan structure. And barrel form for others as a result of wrapped around beta-pleated sheet. So, tandaan, ang alpha helix for support, okay, dahil doon sa kanyang spiral tube structure. Ang beta-pleated sheet naman for rigidness of the protein or for framework, okay. Pwede siyang dalawang structure. You have the fan structure. Example niya is itong sa carboxypeptidase, or pwede rin siyang barrel form, okay, or barrel structure kapag when you use it as a wrap around, okay, binalot mo ng beta-pleated sheet yung iyong protein. So, often, beta-pleated sheet act as the core of a protein with alpha helixes on the outside. Katulad nito, di ba? Nasa core siya, nasa gitna. And then, meron siyang mga alpha helixes na nakapalibot for support. So, a large number of globular proteins are found in larger proteins. Okay? Usually, ang globular proteins mo, nakikita siya usually sa mga malalaking types ng proteins. So, these units, which are the same size of myoglobin and carboxypeptidase A, are called protein domains. Yung mga units, or when you say units ng larger proteins na kasing laki ng myoglobin and carboxypeptidase, ang tawag sa kanila is protein domains. So, a larger protein is like many myoglobin-sized proteins connected together by peptide bonds. Okay? Yung mas malalaki pa sa myoglobins, para silang kumpul-kumpul ng myoglobin-sized proteins connected by your different peptide bonds. So, each protein domain has its own secondary structure that has its task in the protein overall function. So, ito yung example natin ng ribbon structure. Okay? So, sa carboxypeptidase ito. Ang space filling model niya yan. So, parang mahirap siya intindihin. Malabo kasi class, pero parang kumpul-kumpul lang siya anyway. Same lang din doon sa kanina, di ba? Mas specific yung example na ribbon structure. So, ayan siya. Di ba? Meron siyang core na beta-pleated sheet. Okay? Flat na may arrows yung sa gitna niya. And then sa outside niya, used for support are the helixes, alpha helixes. So, yan yung ribbon structure ng inyong carboxypeptidase. Okay, so dyan lang muna tayo mag-end for now. That is the part 1 for your protein or chapter 5, amino acids, peptides, and proteins. So, next Wednesday naman yung part 2. So, thank you for watching. I hope na may natutunan kayo, class. I will upload this and sana ma-upload dito. Mahina kasi yung internet namin. Okay, so see you on the next video and study well.